Good morning, good afternoon, good evening, wherever in the world you are joining us from. I'm Jarka Glassey. I'm the Vice President Executive of the European Federation of Chemical Engineering, and I'm currently in my second term as the Vice President. I graduated as a biochemical engineer from the Slovak Technical University in Bratislava in Slovakia, and until recently I worked as a professor in Newcastle University in the UK, where I actually worked a lot with pharmaceutical and biopharmaceutical industries, and monoclonal antibodies definitely very much of interest to me. And thus I am very happy to be involved in the introduction of this webinar on crystallizing the future from small biomolecules to monoclonal antibodies, and I'm really looking forward to participating in it as much as I can. Let me first say something about the Spotlight Talk series. This is now our sixth year of the Spotlight Talk initiative, which started in 2020 to help us keep together the community during the COVID pandemic. The success of the previous years convinced us to transform the spotlights into a regular feature of the EFCE program. And we now have one spotlight series in odd years, like 2025, and two in even years. This spring, 11 of our working parties and sections are delivering over 2 weeks, or have delivered over the 2 weeks, webinars with talks on specific topics by leading industrial and academic experts. I'm saying have delivered because I think today is the last day. So if you've missed any of the webinars you were interested in, you would need to have a look at our recordings once they are available. The series also enables attendees to sample matters in new areas, encouraging cross-fertilization of interest. And as I said, the complete program is available on EFCE website, and you will be able to access recordings as well. EFCE promotes scientific collaboration and supports the work of chemical engineers in 30 European countries, representing more than 100,000 chemical engineers in Europe. EFCE's working parties and sections cover all major specific aspects of chemical engineering and are in fact at the core of our organization, forming the scientific engine that drives many of our activities. They provide an important forum for technical exchange and networking amongst chemical engineers in Europe. I would really like to encourage you, if you're not already involved with some of our working parties and sections, to consider joining. The section membership is open to colleagues working in the area for individual membership, although for working party membership, delegates are either nominated by their national member society or elected by the working party. If you are interested in joining, do reach out to us through our website or your national member society. Before concluding, I would like to thank all the people who worked hard inside the working parties, sections, and in the EFC in general for this initiative to happen and to be successful. And in particular, many thanks to our EFC's Martin Pu in Toulouse, who did most of the conceptual setting up and practical activities. As always, Martin, really appreciated. I'm also pleased to inform you that the EFCE board has decided to award to Martine the prestigious Dieter Behrens Medal for her contribution to chemical engineering that will be delivered at the EASE ECAP conference in Lisbon in September. So thanks again, Martine, and congratulations on this very well-deserved award. Thank you for your attention, and I wish all the speakers and the attendees a fruitful and successful webinar. And I would like to now hand back to Elena Simone from Polytechnico di Torino in Italy to start the proceedings. Good luck, everybody.
Thanks, Jarka, for the presentation, and welcome everyone to the Spotlight Talk organized by the Working Party of Crystallization of the European Federation of Chemical Engineering. I'm Elena, and I'm a professor of chemical engineering in Torino. I just have a very brief presentation before we leave the stage to our speaker. So I'm going to share my screen, and I hope you can all see my screen and my presentation. So this is the plan for today. We have three talks from three different scientists in different stages of their career. And a little bit different geographical origin. All the talks are focused on relatively complex crystallization processes. So we're going to deal with peptide crystallization and protein crystallization. So new types of therapeutics that are becoming more and more important, together with still with the small molecules. And then we have also a third presentation, in this case from an industrial scientist, which is still focused maybe on more traditional pharmaceutical molecules, but we're still dealing with more and more complex processes where we have different components. So I think the common area here is difficult crystallization processes in complex media, which can be also biocrystallization. Just a suggestion and a notice for the participants: you're not allowed to use your microphone, so please use the Q&A section for question, which we're going to read, me and my colleague Burak Eral from the University of Delft, my co-chair, during the presentation. Since I have just a few minutes, I just want to share some slides that were provided to me by our chair of the Working Party of Crystallization, just to briefly summarize what we do and who we are. So the Working Party is let's remove this. Now have 22 members, and for each member state we have two delegates, one for the industrial part and one for the academia. We also have elected members, so myself and Burak, we are elected member from different countries, also outside Europe. What we do is we try to foster collaboration and stimulate discussion in the topic of crystallization. We are a diverse community that work on different problems, but when we think about crystallization, the fundamentals are the same, and many problems are the same, even though we work with different compounds. We organize events, so our main event is the International Symposium of Industrial Crystallization. So the last one was in 2023 in Glasgow, but we're currently organizing the in Budapest in 2026, so stay tuned because we would like people to participate. And then we also organize the spotlight talk. So we have one in machine learning and one we joined with the particle characterization party on innovative characterization technique for particulate and crystallization processes. If you've missed them, go on YouTube and you can watch them. We also organize other events. So we just had a workshop in September last year with several people from the Working Party, and also we offer summer school to early career researchers. So the next is the last was in 2024, and they're normally held every 2 years. So again, next year we will try and welcome you. The Working Party also has its own award, the Excellence Award in Crystallization. I was lucky to receive this award in 2017, and the last two people winning it were Dr. Ashwin Rajagopalan from the University of Manchester and Francesca Breveieri from ETH Zurich. So again, close to the ISIC, we normally offer the opportunity for nomination for these awards. So if you have done a PhD in crystallization, please have a look at our website for the rules for nomination. So I am done with my presentation, so I think now I can leave the stage to our first speaker, Professor Jerry Heng. He's professor in chemical engineering at Imperial College London, and he did his PhD there, and he's been working on crystallization for several years, looking at surfaces and the effect of crystallization on surfaces and characterization of surfaces. And now recently, now it's been a few years actually. He's been working on biomolecules and biopharmaceutical crystallization, particularly peptides. So the word to you, Jerry, and thanks for presenting.
Thanks very much, Elena. Good morning, good afternoon, good evening to the attendees. I have to say a huge thank you also to the organizers, of course, Elena and Burak for the kind invitation, to Martin for coordinating everything, Jarka for the fantastic talk about what EFCE does. And it's a great honor and pleasure for me to share today's session together with Joana and also with Fredrik. First, disclaimer already, it's my great pleasure to share with you some of the work around peptide crystallization that the group does, but really it's the hard work and the excellent work from very many scientists and researchers in the group. And I hope I will not forget to mention their names at the right slide. And if I do, I hope to capture them at the acknowledgement slides later on. But just as a means of providing some background around peptide crystallization, you would know that peptides are made of amino acids put together from the amide groups, and they're being increasingly important over the last couple of decades, primarily with some of these blockbuster drugs being in the news, the likes of your liraglutides and semaglutides. But the peptides has quite a number of unique features. Amongst others, the peptides do have fatty acids to enhance delivery. Peptides are synthesized via solid phase peptide synthesis route, normally on a bottoms-up approach via deprotections and protecting and deprotection steps. Peptides can be, I might have mentioned, cyclic in nature, and we'll come back to this case of a peptide protein being insulin with its disulfide bonds across them, making it one of the only biopharmaceutical which is purified, isolated, purified, manufactured, and delivered in the crystalline form. Peptides are not only have a range of therapeutic effects, amongst others, as you can see here, ranging from oncology, cardiovascular, autoimmune disorders, and the likes of peptides, which are quite common in the news at the moment, the GLP-1, GLP, analog peptides, perhaps the names Ozempic and Mounjaro may trigger some relevance to or some recognitions to you. The peptide manufacturing, typically, if we run through what Hamish Mitchell has provided around these aspects of synthesis, purification, and formulation, it is that purification step that we as a chemical engineer are interested in trying to drive some improvements, because the current traditional approaches of chromatography are often slow, expensive, and potentially also quite not tremendously environmentally friendly with the use of large amounts of solvents. That remains the bottleneck in the manufacturing and delivery of peptides to patients. So the question about this bottleneck is, how could we then alleviate that manufacturing problem? As I mentioned, quite a large amounts of the downstream costs is associated to chromatographic purification routes, and hence what we try to do in the group in our research lab at Imperial College in London is to develop three-dimensional nanotemplate approaches for direct isolation purification from the likes of, say, a fermentation broth in the case of proteins, or for the purification of peptides. So the idea around peptide crystallization is that there are several benefits. Benefits range from use of less solvents, more energy efficient, ability for impurity rejection, through to enhanced stability or properties of our therapeutics. There are challenges. Of course, peptides are inherently quite flexible. They could be quite large, anywhere between 2 to 50 amino acids typically are assumed to be in that peptide categorization. And of course, that flexibility means that a molecule may not necessarily like to be ordered and be arranged in a crystalline structure. So the aims of the talk this morning, over the next 25 minutes, I have got to include some time for question and answers, of course, is to try to demonstrate the applicability of crystallization of peptides, to develop some aspects of workflow, and to show results, of course, at the end of the day, around some of the strategies that we've deployed for optimization of peptide crystallization. If time does permit, I may flash through a couple of slides as well on computational approaches in that process design scale-up approach. We first started this adventure really looking at phase diagram, trying to understand how solubility of peptides change over temperature, over solvents. And Mingxia Guo, who is no longer at MIT now, she was a PhD postdoc at Imperial with us, went over to the US, and I think she's working in industry at the moment, was to measure the solubility of what we term as the homopeptide series here. Simply peptides made of glycine, 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 glycine in the di, tri, tetra, penta, hexa peptide sequence. The reason in doing this is for us to now try to build in solubility models by understanding the contribution of the site groups. In this case, of course, glycine only has a residual group of H. One could imagine then in such a generation of solubility properties, we could build up a whole range exemplified here by glycine phenylalanine, glycine thyroxine, glycine aspartic acid, or glycine histidine, and building up sort of the databases to understand solubilities of such materials. So of course, Mingxia painstakingly measured them to try to understand phase diagrams and stability and properties of peptides. And we stumbled upon a I wouldn't say problem or issue, but one of the first things that we wanted to ponder here was simply the effect of sequence. The effect of sequence really is just the order of the amino acid groups. And Shu Liang, who was the PhD student, and he's gone off to work in industry in AstraZeneca at the moment, was looking at the solubility of just simple dipeptides. So in these gray squares are aniline anilines, and you see the solubility as a function of temperature given by these gray squares, compared to the solubility of glycine, these blue diamonds, the solubility is a function of temperature. I would have imagined, if I were to guess simply based on weighted fractions, that if I've got a dipeptide made of aniline and glycine, or glycine and aniline, that they will fall between the gray and the blue lines, and they did not, right? So the glycine aniline solubility was right at the top, and the aniline glycine in green here is right at the bottom. So one could not really just predict simply its solubility directly from the composition of the constituent amino acid within the case, or within this dipeptide case. It really goes back, I think, to its crystal structure, the need to know the likes of its thermodynamic properties from its melting temperature to latent heat of fusion. So this effect of sequence exemplifies that it is absolutely important for us not only to know the groups that make up the peptide, but the order of sequence, what order these amino acids are based in. Looking at manufacturing, jumping on quite quickly through these various elements in peptide manufacturing, Jianing Li, a current PhD student in the group, is looking at the effect of the protecting groups. So we do know that we use protecting groups in peptide synthesis so that the reactions, if you like, the amide bonds are formed in the correct places. And these protecting groups are typically the likes of your FMOC, BOC, or CBZ chemistry. Just to demonstrate here that in green, these are the solubilities of aniline, and in purple, this is the solubility of aniline with a protecting chemistry group BOC. And already one could see that the solubility is tremendously reduced with the presence of a BOC chemistry. The importance here, or the relevance, is in the workflow development later on, as we could try to develop strategies to crystallize or isolate molecules out or to crystallize molecules out with or without the protecting groups. And in our experience, obviously the protecting groups here play a large role in the crystallizability of the molecule. All these experiments are very painstaking and takes quite an enormous amount of time, and we can't possibly do all the experiments 24/7 because we do close our labs. I know they may allow you in a little bit earlier at MIT, as Joana mentioned in our pre-session, but Imperial students are not allowed to stay 24/7, or researchers are not allowed to stay 24 hours a day in the lab. But we use automated approaches. Isha Bade, a postdoc in the group at the moment, has developed these sort of automated approaches using a ChemSpeed type of system. I failed to mention that there's a dosing unit measuring small amounts of solids into the vials, followed on by addition of liquids. You might have just seen that the stage can be stirred. The vials are placed in a block to control temperatures. I would suggest potentially in this case it's easier to vary your solvents rather than varying temperatures across the block. Liquids or the solvents there are then filtered, and then we could obviously measure by some means of spectroscopic methods to determine solubility within the solution. The idea and the aim here is not only to get more controlled experiments and hopefully more precise, making less mistakes, but also to generate phase diagram or solubility diagrams for new peptides much more quickly. I would imagine in the order of at least 10 to 100 times faster than manual measurements. We did stumble in the case of dark triglycine, one of the first peculiar occurrence in these systems, in that the work that Ming-Xia was conducting resulted in habits that we did not see before. These crystals were fortunately large enough, and so we took them across to our chemistry department to run single X-ray, single crystal XRD. And in those single crystal XRD, we were able to solve the structure for this triglycine. And this triglycine did have two water molecules, so it's a triglycine dihydrate. And we tried to deposit it, or we tried to search in the Cambridge crystallographic database, and there was no such entry for triglycine with the dihydrate system. So in this case, we found a new crystal structure, but most interestingly, this triglycine adopted a different conformation to its anhydrous form. The anhydrous form was linear, but the dihydrate form started to adopt a PP2 conformation, demonstrating already changes in its molecular conformation and crystal packing, even at short-chain peptides. We extended the work here to look at the effect of salts and temperatures to understand stability of the hydrate-anhydrous systems, and it goes quite well at higher temperatures. The anhydrous form will be present, whereas at lower temperatures, of course, the dehydrated form will be present. We were looking at the effect of residual groups on the crystallization kinetics here. So just measuring the probability for the case of glycine, diglycine, and triglycine as a function of time to obtain some degree of kinetic parameters for simple homopeptides. And I guess it's not surprising that with increasing residual groups, so the number going from di to tri to tetra to penta, the induction time increases quite significantly. What Vivek Verma did, and in the drive to try to control nucleation, was to compare homogeneous to heterogeneous nucleation behaviors. You will see here at a certain degree of subsaturation, 1.44, the induction time for an unseeded case, we call an unseeded case in a batch crystallizer being homogeneous nucleation, whereas the one with seeds, and the seeds we use here are three-dimensional nanotemplates made of silica, that the induction time has reduced quite significantly, right? From about 150 minutes or thereabouts to about 40 minutes or thereabouts. So this is the case of triglycine dihydrate, triglycine. And it was not limited to triglycine. If you look at the table here, in all cases of gly and digly and trigly that Vivek tested, the homogeneous or the seeded cases was always much shorter in induction time than the cases for homogeneous nucleation. Some calculations, some fit to the classical nucleation theory type of models suggest that at such low concentrations, the effect of seeding, the heterogeneous nucleation, did not alter much our interfacial free energy, gamma or sigma in the typical type of classical nucleation equations that you may be familiar with. It did, however, change the pre-exponential function for the case of homopeptides. We tested, of course, here with various types of templates. We could, like, if you like, we could engineer the templates to have different porosities. We tested porosities from 6 to 10 to 30 to 50 nanometers. We could, of course, derivatize these pores as well with different chemical moieties. In this case that I'm showing on this slide for the experiment using diglycine, they were just hydroxyl terminated groups. And the effect of pore here could be quite distinct, with the 6 nanometer templates resulting in the lowest induction time demonstrated in this red trend line, with the black trend line being a homogeneous case. And Shu Liang continued on his work to look at the effect of impurities. Look forward to Fredrik's talk later on the complexities of solutions. In this case, we were trying to crystallize diglycine, pretending in the solution that there was some impurity, for example, in an error deletion synthesis approach. If glycine did not react to form diglycine, we have glycine left over in the solution. What would that do to our crystallization process? It did not seem to have a large impact on the solubility. So the solubility of diglycine remained almost constant with increasing amounts of glycine in the concentration, but it did have a large impact on the induction time. You see that this black trend line is just the desupersaturation profile for diglycine with no impurity, red with 2.5% of glycine, and already the induction time has increased quite significantly. But at 10% of us, this would be a very high degree of impurities. The induction time increased from a couple of hours to over 15 hours. So small amounts of impurities do impact on the crystallization behavior, the nucleation and induction times. But we again did see something peculiar. So in the case of triglycine, previously I mentioned we saw a new habit, a new polymorph of triglycine dihydrate. In this case, what happened was we again found a new co-crystal system. This co-crystal system is a co-crystal of diglycine with glycine, and also with the presence of water. So glycine, diglycine hydrate. And what Anshu built here is a phase diagram, a ternary phase diagram, to show where this co-crystal hydrate is favored. So there are conditions at which we may drive only the diglycine to crystallize out, the glycine to crystallize out, or a hot or a sweet spot where the co-crystal hydrate form of digly will preferentially crystallize out. So I've only been showing you results of very simple homopeptides of glycines or anilines, and you may ask, how about a slightly more complex real peptide? This is work of Vivek Verma looking at a real peptide now for cancer, for a cancer treatment type of peptide of 10 amino acid long, and these 4 peptides, doralins, gosrelins, buserelins, and leuprolides, really only differ very, very minimally. So for example, buserelins and leuprolides are identical in position 1, 2, 3, 4, 5, 7, 8, 9, 10. They only differ in position 6. So only one of the residual site group differs, right? And the same happens, for example, if I look at gosrelins and buserelins, they're identical in 1, 2, 3, 4, 5, 6, 7, 8, 9, and only differ in position 10, right? And that screening conditions is quite tricky. We've spent over 2 years, and we only were able to crystallize out one of these 4 leuprolides in a specific condition as reported here, confirming that this 10 amino acid long peptide is indeed, this crystal is indeed a peptide crystal by use of light microscopy or bright field UV microscopy. So we're quite happy there, although we've not quite been able to solve this crystal structure quite yet. Fredrik Link, he's now in consulting in Switzerland, worked on insulin, and in the case of insulin, we wanted to try to control its nucleation behavior using what we call soft templates. So these are the use of amino acids in solution. They are dissolved in the solvent, just testing on arginine, glycines, leucine. So the higher the bar, for example, the higher the green bar, demonstrates that increasing amounts of leucine, the probability of the occurrence of insulin crystallizing our solution also increased. Um, what Fredrik was working on was to test out different pH conditions, and obviously here we demonstrate that pH was quite, or insulin crystallization was quite sensitive to pH, and Fredrik has also been able to transfer this into a slightly larger process, not only at milliliters or microliters in scale, but into a somewhat continuous flow crystallization, where we demonstrate that the pre-mixing chamber is quite important in the crystallizability and crystallization profile of insulin. I sidetracked a little bit. I know, Jarka, you mentioned about monoclonal antibodies. This isn't a peptide. This is a monoclonal antibody work that Hua Yu Yang at Loughborough University now and Wen Chen Chen at National University in Singapore via a European Union FET Open program together with collaborators across Europe, driven out from Italy, CNR ITM in Calabria. So we were looking at this drug, anti-CD20, which is rituximab, and spending quite a while before we were able to obtain some crystals out of the solution. This was from level 1. So level 1 really meant that anti-CD20 was expressed upstream, purified via a whole series of chromatographic steps. So level 1 is a very pure form. And if we were able to crystallize it, that was great, of course, but we did not reduce the manufacturing cost of it. So we spent a little while, I'm going to jump through that slide. We spent a little while, but input from collaborators, and I do appreciate a lot of work that excellent modelers and computational scientists have guided our experimental work. This is work from Jim Lutsko and Julian Lam, who demonstrated a certain specific pore dimension was ideal for the nucleation or crystallization of anti-CD20. We went about doing that. So we drilled holes. These holes were 60 nanometers. We called them Imperial 650. The key result here is that we're able then to crystallize anti-CD20 out of level 3. So out of level 3 means that the solution that we started off was really just post DEF filtration without any protein A or anion exchange or cation exchange filtration. So those were not pre-purified or pre-concentrated solutions. If I would imagine that costs in downstream, there would be a significant reduction in cost if one could crystallize out from level 3, as we exemplify here in the case of anti-CD20. I will go back to peptides. This is work that Hamish Mitchell, he has graduated and he's joined UCL just down the road, across the road from Imperial on a fellowship. But Hamish has been working with Eli Lilly on a compound called GG353. GG353 is quite a large, complex a molecule, about 39 amino acid. Hamish is on the call, so if I can't answer any difficult questions, Hamish is on standby, of course. But it also does have a very big fatty acid molecule. But what we wanted to do in developing the workflow here is to really look at methodologies around screening within the protein database, developing from small-scale vapor diffusion, sparse matrix screening, through to obtaining some successful crystallization. I wouldn't be here today if it was not successful to share with you these results of these crystals are that of the 39 mer, meaning 39 amino acid large GG353 protein with a fatty acid side group. These were stained blue to prove that they are not salt crystals. We were very fortunate to work with Eli Lilly, that's Emily Ginn and others from the Lilly side on an APSRC Prosperity Partnership project. Now, of course, being in an engineering department, one often thinks about scaling things up, and what Hamish have been working on in scaling up posts crystal index screening, crystal hit in the index screen, is to try to optimize further the vapor diffusion process, develop microseeding development approaches, transitioning microbatch to larger scale crystallization experiments. And what this has resulted in, if I could jump, I think I would jump this slide because I'm starting to run out of time, is the ability through microseeding techniques for us to obtain, us being Hamish and others, to obtain very, very nice crystals very nice in this case, meaning that they do diffract X-rays well. So what microseeding matrix has been able to achieve was to overcome poor crystal properties. And what Hamish and colleagues at Lilly have been able to do is to now define and resolve the crystal structure for that 39 amino acid long peptide with a fatty acid side group to a resolution of 1.7 angstroms. I believe this would be probably one of the first peptides with fatty acid side group crystallized out and solved to quite a reasonable high resolution. Just two more slides before I wrap up. So the use of experiments with modeling, we employ GPROMs as a method to help us investigate applicability of some of the models around nucleation and growth to describe crystallization processes. In scaled up experiments, we first tested these on insulin and lysozyme, including the use of seeds. We use glass beads, silica particles to control and enhance nucleation tendencies. I think the key thing is that one is able to describe nucleation processes quite accurately, and the models do fit reasonably well. I would not want to generalize this to say that it fits perfectly for every type of large molecular system that you have. There are always going to be some improvements to the model, but what we've seen so far with the case of lysozyme and insulin, we're able to compare experimental data to models obtained quite in good agreement. I am at my last slide. I'm looking at my clock. I've got 3-4 minutes left, I think, to the 30 minutes that is. So in conclusion, I hope I've given you a very quick tour of some of the activities that we've done around the use of surfaces to control nucleation crystallization. And I think that crystallization is indeed a viable route for the purification, isolation, purification of complex molecules, including that of proteins and peptides. Scaling up, work to do still, but not impossible. I'm a believer I'm an experimentalist by training, but I'm a believer in the synergy between modeling and experiments, as models can help guide experiments, and hopefully experiments can also have input into the modeling ability. There's a lot of exciting things I would argue in peptide crystallization, from discovery of new polymorphic forms, hydrates, solvates, co-crystals. There's still a lot of surprises that is being yet to be discovered. So with this, I like to plug in a shout out, because there's still quite a lot of activity in this area. We've been very fortunate with a program that's led by Professor Jose Teixeira, with Dr. Felipe Castro from the University of Minho, leading on a Marie Sklodowska-Curie action doctoral network called Pro Crystals. You may recognize some of the very many names here within the EFCE network in crystallization too, that Elena might have mentioned already. So please do watch this space. And with that, I'd like to thank the researchers. I wouldn't be able to name all their names this morning, afternoon, but thank you very much for your kind attention.
Thanks, Jerry, for the nice presentation. If any participant have a question, please use the Q&A section, which I see there's no question at the moment. I don't know if, Jerry, do you see any? And I'm looking at the...
No, I'm also monitoring. There are no questions at the moment, but
okay.
Yeah, if I may, I might steer the pot a little bit.
Sure, go ahead.
Yes. Hi, Jerry. Again, thank you very much for a nice question. So in different industries, what a successful crystallization looks like is quite different. How is it for peptide crystallization?
And no, I think you're absolutely right, Burak. The key performance indicators for any process is different, even in not only in industry, right? In academia, if I was a structural biologist, I only need one crystal high X-ray diffracting to 1.5 or 2 angstroms, and I'll be happy. As an engineer, I would say I want a billion particles, if not many trillion and billions of particles from a process. So I think our key performance indicators would be driven by what the need would be to produce a product at a certain quantity within a certain time frame of a specified purity. And we can up that challenge from less pure solutions, faster, greater yields. So I guess our key performance indicators would be derived based on what our key objectives are as well at various steps. Yeah. Sorry, Burak, I'm not giving you a direct answer to a question, but I think it does evolve at different stages.
Exactly. I think it is, if I understand your question correctly, it's very much dependent on the manufacturing demands at that moment.
Well, beyond that, Burak, I hope not to take too much of Joana's time next, but let me put it this way too. I think there are multiple drivers from manufacturing, cost efficiencies, sustainability, patient friendliness, efficacy, and I think buyers, of course, from my view, I would argue that peptide crystallization could offer quite a lot of solutions that we just to the challenges that we just mentioned just now.
Thank you very much.
Now I see a lot of question. So guys, participant, please keep asking question because Jerry's gonna stay on the call and he can answer on the chat. But maybe you can just reply to the first one from Laurent Cassaire. Did you implement thermodynamic modeling to represent the solubility of the various peptides?
Yeah, no, thank you very much, Lauren, for the question on thermodynamic modeling for solubility. We did. We worked with Professor Amparo Galindo, George Jackson, using the gamma SAF, that's the statistical associated fluid thermodynamics model. And I think more recently, I forgot his name now, had just published a piece of work in IECR on modeling of such solubilities for relatively simple short chain peptides, so less than 10.5 or less sort of level. So yes is the short answer. And you're right, Elena, I will try to respond to a couple of other questions over there, which I just see as well too.
Thanks a lot, Jerry, and thanks for your presentation again. So I'll just introduce the next speaker, Dr. Joana Ferreira. She's currently a postdoc at MIT, but she did her study, she did a joint PhD at the University of Porto and KU Leuven. She's been working on protein crystallization, and she has a very interesting approach to the problem, looking at both modeling and experimental. She did quite nice work for her PhD thesis. So the stage to you, Joana. And thanks for waking up so early this morning.
Thank you, Elena. So I will share my screen. So good morning, good afternoon, good evening, everyone. My name is Joana Ferreira, and thank you, Elena, for the nice introduction. You saved me a couple of minutes, I would say. And for this talk today, I will be focused on protein crystallization. And I thought about dividing my talk in 4 parts. So I called it 4D. So we have size, scale, methodology, and analysis. And I'm currently a postdoc at the Meyerson Research Group at MIT, and I will mostly focus on the current work that I've been doing for the past year since I joined Meyerson Group. So while deciding about my title, I thought about why not dividing these, let's say, in 4 pieces of a puzzle. So we have size, scale, analysis, and methodology, and I will try to go through each of those. So in terms of size, we can consider the complex nature of proteins, and it can vary from a very simple amino acid. We have glycine here as the example. This is the 3D structure of glycine, and Jerry already talked a lot about it. Then we can go through model proteins. I have here the structure of lysozyme and insulin, two proteins that are very familiar to me because I did a PhD about them. And then we can go through large molecules. And here is a 3D structure of a monoclonal antibody. And my talk today is going to be mostly focused on these very complex proteins, monoclonal antibodies. In terms of scale, we can think about very easy experiments using a hanging drop or sitting drop. We can save a lot of material, and is always a nice technique to start if you have no idea about your crystallization conditions. And we can go to a large scale, thinking about scale-up strategies, and then even going further and thinking about the continuous process. And during my talk today, I will also focus a bit on our current developments with the continuous experimentation for monoclonal antibodies. And then the other two pieces of the puzzle, analysis and methodology, I think they should always be together. And this will allow us basically to put everything on the same page. We have the molecules, we have different scales, and then how can we make a synergy between the experimental and the computational part. And this is also something that I'm going to cover today. But before going through the scientific part, something that I want to highlight is the current trend in the big pharma, and also especially looking at the American market size. As you can see, there is a clear trend in terms of the mAbs market size through the years, and it is expected that in 5 years we can reach a huge number. And if you compare to 2025, we can almost double in terms of the market size. These are really huge numbers. And if you look at the context also of the big pharma, the ones involved in the current trend of biologics, we can see also that the top selling compounds of, for example, Merck, Janssen & Janssen, and Roche, they are monoclonal antibodies, especially Roche. The 3 top selling compounds are all of them monoclonal antibodies. Another clear example of how biologics are being so trend nowadays is also Keytruda from Merck, that occupies most of the selling market. And in terms of the current mAbs manufacturing pipeline, we start from the cell line development, then we go through the upstream and downstream processing until reaching the final stage, the formulation. However, the downstream processing is currently a bottleneck, and this is mostly because of the high cost of the purification steps, which is something that Jerry already brought to our attention. And this is mostly because up to 70% of the total manufacturing costs are due to the different purification steps. And the most traditional way to purify is using chromatography-based techniques, but this involves high operation costs, and it also has a limited throughput. So we think that crystallization might be a good solution for this problem because it can handle high process volumes, it heals high pure suspensions in a single step, and then also in terms of crystalline formulations, we can deal with very stable formulations with also a long shelf time and also with a target drug specificity. However, before reaching a formulation or having a crystalline formulation, we need to crystallize. And crystallizing a monoclonal antibody or a protein in general is not an easy task. And normally, to do so, we think about this as a kind of a flow chart. So we start with the screening, then we can try to measure some solubilities to gain a further understanding of the process, and then we try to quantify through a kinetics estimation. The screening might already be challenging for these kind of molecules. Our first source of information is from the protein data bank. So if you have the 3D structure reported in the protein data bank, we always have a starting point. However, for monoclonal antibodies, talking about full-length monoclonal antibodies, there is only one entrance in the protein data bank. So we can dig in more in the literature, but there are mostly some old patents. There are not a lot of papers. So as a starting point, we can use screening kits. So we can test a lot of different conditions and try to get some good hints. This might involve thousands and thousands of trials. So we also combine this screening strategy with the molecular modeling and machine learning, because this gives us information in terms of the propensity to crystallize. So with this synergy between the experimental and the simulation side of things, we can gain further understanding, and also it saves us a lot of time and money in the lab. So for the screening part, we start at a very small scale using sitting drop, just less than 1 microliter. Then we go a slightly larger scale using hanging drop. And then our idea, because we want to reach a continuous process, we go through microbatch and then eventually crystallizing in continuous. So here is one example of an outcome from 96 different conditions. And we can run thousands of conditions using, I don't know why my video is not working. So here it should be a video of this high throughput facility that allows us to pipette the protein, mix it with the reservoir solution, and then we can run a lot of different conditions in a single plate. However, most of our outcomes from our crystallization trials are mostly precipitants. As you can see here in this graph, most of the conditions end up with precipitation, and only a few of them gives us crystals. Here I'm showing one condition out of thousands where we could see some needle-shaped crystals. So at this point, we are able to start thinking about scaling up, trying to crystallize at a larger scale. So after this successful outcome, we tried also temperature cycling for one of the maps that we are dealing here with at MIT, because as you could see in the previous graph, it's very easy to precipitate a map. So we thought about temperature cycling as a strategy to suppress the formation of the precipitants while inducing the formation of the crystals. Here is one example. So we do the temperature cycling several times, so different cycles of cooling and heating continuously. And by increasing the number of cycles, we could see that we can suppress the initial precipitation, and at the end we can mostly observe crystals. We can also try to optimize here the highest temperature of the cycle, and here is one example of a high molecular weight of PEG, and then as you can also see, we can really suppress the formation of the precipitants, and at the end observe some nice and large crystals. We could also see the impact on the phase diagram. By increasing the number of cycles, we are close to reach the solubility limit. Here, something that I also want to point out is the fact that this has been reported for more complex proteins, and it is something that we also observe for this specific map, that you can basically divide the phase diagram, the solubility limit, into different regions. So at a high temperature, mostly the formation of crystals, while at a lower temperature, mostly precipitation. So it's the reason you have here a significant drop in the concentration in function of temperature. We also did some time-lapsing experiments. So this case, we mostly observed crystals, and then it was also nice that over time we could estimate some kinetics and observe the nice growth, the nice length increase of the crystals over time. And then for another PEG, it was also nice that through temperature cycling, we could really suppress the formation of the precipitants during the heating step. And then at the end, we could see some nice crystals. This process also gave us a good yield, especially looking at the high molecular weights of PEG, more than 70%, as the case here highlighted in the slide. So at this point, we also started thinking about how we can translate our concept of temperature cycling, a concept that has been applied for many proteins. So it's not we are not discovering the wheel. We are just making use of a very old concept in our favor. So if we think about translating this to a continuous process, so what we did was to combine a series of MSMPRs, and then basically what we are doing is transferring the material between the cold and the hot reactor. So it's the same concept. Try to dissolve the least stable form, the precipitate, here in the hot reactor, and then on the cold reactor we are inducing the formation of crystals. So this is still an ongoing research. What is also nice is because with the reactors that we are designing and developing at MIT, we also have access to different probes. So for example, here the Blaze matrix probe can give us some inline information. And as a proof of concept, we tried this with one of the maps. And if you compare the same time step in the hot or the cold reactor, we can already see that, in the cold reactor, we mostly see precipitants, while in the hot reactor, we can suppress the formation of the precipitants and induce the formation of crystals. The second example that I'm showing, so this is the work, most recent work with a different map. So the same strategy, we start with the high throughput screening. We try to get our first good hints where we can see some crystal formation. Then we start scaling up from sitting drop to micro batch. And then we can think about the continuous process. So for this specific map, we did 3 different runs. So 3 different initial supersaturations, 3 different residence time, but always keeping the same volume reactor, less than 10 ml. And here what it was very interesting is that for the third and last run, we were able to run it for more than a week, 8 days. This is something very, very, very long, and it hasn't been reported for proteins. So in this graph, I'm showing the decrease in the concentration over time for the batch process. So what we do is we start we have this startup, or we can call it equilibrium step, where we just run it. We just run a batch process. We obtain the first seeds, and then we start pumping. So we are running our continuous process, and we do so for 8 residence time. So here you can see the decrease in the concentration for the 3 different runs. And what it is interesting to see is the different time frame if you compare 5 and to 1. So in terms of the setup, we have 3 different MSMPRs. So the first one on the right-hand side is where we run the batch process. Then we transfer the material to the second reactor. And we do this because we have different probes. So on the crystallizer, on the first crystallizer, we have the Blaze probe, where we can get some inline information. Then we transfer the material to the second one, where we have the Raman probe, and then we have our collection vessel. After running the batch step, then we start pumping the material. We have the map combined with the precipitant and the additive. We use PEG and the caffeine. And this is a pressure-driven transfer, and we use this kind of system because the protein crystals are very fragile. So we want to ensure that during the transfer, the crystals don't break. and then we adjust the reactor volume by the deep tube position for the different reactors, and this is how the reactors look like. So if you look at the lid, we can insert different probes, thermopar, the probes that you might have available in your lab. So this is what we do in terms of inline monitoring, but in parallel, we also have offline sampling. So we take samples from both the crystallizer and the collection vessel, and we use microscopy imaging to take a look at how the crystals look like, and then also the concentration using protein A. So this is how the messy setup looks like. It's always nice when you have a messy setup because everything looks more complex. So we have the MAP solution, we have our crystallization solution, the peristaltic pump, and then here you can see the 3 different reactors. So we have our crystallizer, where we have the blaze probe from the bottom. We have the second reactor, where we have the Raman probe also from the bottom, and then the third one, the collection vessel. These reactors have been designed and developed at MIT, so we are interested in optimizing the impeller for especially thinking about the complexity and how fragile the protein crystals are. And then we also have a custom built baffle. The video is not playing, but I wanted to show how we transfer the material from the crystallizer to the second reactor. And here I'm showing the first piece of information that we get with the Blaze and implementing PAT tools. So what is interesting to see here, so we can look at the turbidity and total crystal counts, and as soon as we reach a stable plateau, it means that we reach a steady state. So what means reaching a steady state? It means that you look at your real-time crystal size distributions, and the lines are overlapping because there is no further nucleation or growth taking place. You just reach a steady state. So we compare the 2 different runs, and what is also nice to see here is because of the inline monitoring, we can see how the crystals grow over time for the different cases, and also compare it with the offline sampling using optical microscopy. We also have inline monitoring of Raman, so we can also look at the characteristic peak of this specific map. However, we are always limited in terms of the map concentration, so we could only see it for the first run at higher supersaturation. So for the run at high supersaturation, the 5 milligrams per milliliter, we can see that it takes not a lot of time to observe a drop in the concentration because of the high supersaturation. So the tendency is always to see some aggregated crystals, while for the run at 2 milligrams per milliliter, we see some nicer, more dispersed crystals and longer. But it's also nice that with offline sampling strategy, we can see the tendency of the growth over time. So initially, after the batch run, we tend to see some aggregation, but then over time it gets suppressed because of the drop in the concentration, where we are mostly promoting growth. And then also in the collection vessel, because we are steering and the material is being transferred, there is also the Raman crystallizer in between. We can see that the crystals are slightly smaller because there is still some supersaturation. So we might have some crystal breakage or additional nucleation events in the collection vessel. And then for the lowest supersaturation, for the run that we did for more than a week, here is interesting to see that also in the collection vessel, there is still some supersaturation, so we might also have additional nucleation events or even crystal breakage. But here is very clear that from day 1 to day 3, we observe a clear increase in the crystal size, but then from day 3 to day 8, for both crystallizer and collection vessel, we see that the crystals are getting smaller, and this is probably due to secondary nucleation. It was not so for the other cases, it wasn't as clear as for this case. So we are also interested in quantifying crystal size distribution, and here is even nicer because we have inline data from Blaze and then offline from microscopy. So we thought about using because we collected so many images from optical microscopy. So we thought about what can we do with all these images? How can we make use of all this data? So we started developing a GUI on MATLAB, and the idea is to obtain different crystal size distributions over time and then compare it with the data that we collected with Blaze. So we have been trying to optimize the code as much as possible, playing with the different parameters. Here I'm not gonna go too much in detail, but if you have questions, feel free to ask at the end. So we thought about, since these needles are very long and they might be similar to an ellipse, this is the first part of the process, is to detect as many needles as possible. Then we can also play with other thresholds, binarization. Here you can already see that we are actually able to get a good outcome. It kind of represents very well what we see from the microscopy images. But yeah, this is still ongoing progress, ongoing research, because some of the images we might have issues with light. The light is not uniform, then also brightness, contrast. So we are still trying to optimize the code. And then, so if you still remember, we had the 3 different steps: screening, solubility, and kinetics. This is the last step. So we are trying to combine the steady state MSMPR model with the population balance modeling. So with this approach, we can estimate nucleation and growth rates. Also from the population balance modeling, we can come up with an expression for these 2 different variables that are very important to quantify, especially nucleation rates. And we did our first trials with the data collected during the continuous runs. So as you can see, we obtained very high yields for both the batch and the continuous runs. And then we could, from the simulations, we estimated the nucleation and the growth rates for the different runs. And it was interesting to see, because no values have been reported for monoclonal antibodies, but it was interesting to see that these values fall very well with what has been reported for model proteins such as lysozyme and insulin. So it's very nice that we observe some nice crystals, but we still need to ensure that those are in fact protein crystals. So most of the labs can use polarized light. However, the polarized light, the crystals might still be shiny, but they might be salt crystals. So the second step is to use UV light. This gives us direct information if we have or not a protein crystal. We also have tried XRD. However, with the in-house source that we have access here at MIT, we could only we could only see that we don't have salt crystals, but it was not possible to obtain a diffraction pattern. We also tried the cryo-TEM to observe in a more clear way our needle-shaped crystals. Optical microscopy that we used a lot during our continuous runs, Then we also tried the SEM, because for some of the runs, we could see that besides these very needle-shaped crystals, we could also see some plates, and we wanted to take a further look. We also ran SACs because it was in terms of material, we don't need a lot of material to run SACs, and we were very successful. We could see the 4 different peaks for this map that was in accordance with a paper published by Merck very recently with the same map. So we could confirm that in fact we have map crystals. And then at the end, we can also wash our crystals, dissolve them, and run size exclusion chromatography to confirm that, again, we have our map in solution. So all the work that I've presented so far, it was with pure material, pure solutions of maps. But at this point, because it was also one of the goals of the project, was to try to use crystallization as a purification strategy, starting from harvest material. So we got in touch with our colleagues from CBI. They provided us some cell line harvest material. And here the only purification steps that they do is to, in the initial step, through centrifugation, they remove the cells, and then in the second step, they remove the dead cells also using centrifugation. We ran some bioanalytical characterization through protein A and size exclusion chromatography. And what we can see, we see our characteristic peak of the map for a retention time around 1 minute using size exclusion chromatography, and a retention time around 20 minutes from protein A chromatography. And then we can also see the other peaks that are characteristic of impurities. We have many things in solution, exosomes, other small proteins, and some kind of biomaterial based, also DNA. So our initial purification strategy, as soon as we receive this material, because as you can see from the chromatograms, there is a lot more than simply the map. So we use TFF because it was very efficient, removing most of the impurities, and at the same time we are also concentrating the solution. But this was not enough to remove all the impurities. So we also used a DNA depletion protocol that is mostly precipitation, and then an additional precipitation step. Then with all the optimization in terms of the initial purification strategies, we crystallized. We need to adjust the pH of the biomaterial, and then we add the same crystallization solution that we added previously, PEG and caffeine. And then at the end, we can characterize using different analytics, size exclusion chromatography, protein A, BCA, circular dichroism, and so on. And also the solid state characterization, as we did before with the pure material, with SACs, SEM, and optical microscopy. So here is one of the examples, the crystals that we obtain from after purifying the biomaterial. We could see some needle-shaped crystals similar to what we observed with the biomaterial. Then we also used the SEM. We could again see the needle-shaped crystals, also some plates. And then we did run again SACs, and we could again see the 4 characteristic peaks of this map. The scattering is not as, the curves are not as smooth as with the pure material, but we can see the 4 characteristic points, what is in fact the most important part for us. So at this point, this was a very successful outcome, because we could crystallize from harvest material, and this actually opens up very nice and new routes through selective crystallization. For example, what happens if you mix 2 different monoclonal antibodies, like which one is gonna crystallize? And then of course, thinking about the next step, the batch to continuous transition, because all this part with the harvest material, all the experiments were done at a micro batch scale. In terms of bioanalytics, it was also nice to see at the beginning we have the characteristic peaks of the impurities. Then when we mix it with our map, we see the characteristic peak of the map. Then after dissolving the crystals, we still see the peaks of the impurities. But then if you do buffer exchange, we only see the peak of our map for a retention time between 20 and 25 minutes. So we can in fact use crystallization as a purification strategy. We can remove host cell proteins and other impurities, and this is a very sustainable route because at the end of a continuous process we obtain a large volume of crystalline of crystals, so we can wash the crystals, dissolve them, and then start a new process. So this is the reason it's a very sustainable route. We also took a look at circular dichroism to see if we could see any change in terms of the secondary structure. And what we observed, if we compare pure crystals or dissolved crystals from the biomaterial, we could see that this is both of them are characteristic of beta sheet. It's a relatively flat ellipsity, and we can conclude that we have mostly monomers in solution, and we don't see any secondary structure change over time. So at this point, we can see that we can actually combine the 4 pieces of the puzzle. So we have the size, scale, methodology, and analysis. And at this point, I would like to finish my talk. I would just like to thank especially FDA for funding the project, then also the many different groups that have been collaborating together, Myers and Bratz and Trout, also some of the researchers that helped me more directly in this project, also the Europe students who have been working a lot in the lab. And then here just showing the picture of our group. We have new faces who are still not here. Some of them already left. So I would also like to thank you for your attention, and I hope you have questions.
Thanks a lot, Joana. Very nice presentation. There's a few questions on the chat. We're running a bit out of time, but I can maybe you can answer a very quick one from Kerstin Volgemuth from TU Dortmund. Is the transfer between the different vessel intermittent or continuous? I think she's referring to the multiple MSMPR setup, right?
Yes, this would be nicer if the video would actually work because you could really see. But it is intermittent, so we do the transferring every certain amount of time. So depending on the residence time or the total duration of the process, we impose we basically define a transferring time.
Okay, thank you. And yeah, there's a few more questions that maybe you can answer directly on the chat. And yeah, thanks again, Joanna.
Thank you.
And thanks also for again waking up so early. Okay, Burak, do you want to introduce the
Yes.
next speaker?
Yes, as also same goes for Fredrik, the next speaker. Thank you very much for waking up so early to talk to us. So Fredrik did his PhD in KTH in Sweden with Öke Rasmussen. And after that, he followed the industrial path, but he has been an active member of the AIG separations cluster. And he's going to tell us about what he has been doing recently. The floor is yours, Fredrik.
Okay, thank you very much. And yeah, I'm not exactly a morning person here, but I'll try to do my best here in terms of the presentation that's coming. Let's see here. So I should be here. All right. There we go. Sorry. switch.
Display settings.
Yeah.
Right above your curve.
Okay.
Yeah, there.
Oh,
Next,
there we go.
next. One next.
Yeah, it just gets this audio. Let's see if I can I move this out? Oh, there we go. There we go. Okay. All right. So thank you so much for inviting me to this talk. And yeah, so my name is Fred Nordstrom. So I work at a pharmaceutical company called Boehringer-Ingelheim. It's a German company. It's a private company. So it's very unusual. And yeah, today I will talk about crystallization that we do in multi-component media. And this relates to not the proteins or the peptides you've seen before, but more of the small molecule, even though some of the small molecules nowadays are turning into much more complex and very complicated structures. But I think when it comes to crystallization in industrial settings, everything is crystallization in multi-component media. It's just a question of having more or less of the foreign species or impurities that are present in solution as you crystallize in this. And let me start by going into a study that we did together with another pharmaceutical company, Merck, and also Rowan University. This is back in 2023. And the whole goal of this study was to look into what actually governs the rejection of impurities. So there was a lot of very few studies that were looking into the effect of exactly how crystallization works in terms of how do we reject impurities. It's been very empirical in the past. So we've been dedicating a lot of effort here and try to understand what exactly happens. And what we see here is that most of the impurities that are present in solutions, and these are, sorry, these are, go back, primarily process impurities are structurally similar. So they're like, reagents, degradation products, a lot of different types of impurity have some sort of structural similarity to the API. They actually form these solid solutions with the API, and that's why they're so difficult to reject. And so we collected a total of about 52 examples that we collected from the three different parties here. And about almost 80% or so actually has something to do with solid solution. So either you're forming one solid solution phase, or even sometimes two or multiple. So every time we crystallize something in multi-component media, that inevitably then leads to that we have some sort of solid solution formation. And me personally, I had very little understanding about solid solutions starting out. So I tried to learn more. And it's funny because when you're in the organic sort of realm of the sciences, the whole concept of solid state miscibility and solid solutions is very limited. But if you go into other fields, I have another slide here, show this. If you go into geology, mineralogy, people working with atoms like steel or different types of elements and alloys or that sort of things, then yeah, they have tons of experience and they have collected that over maybe 100 years or so. So I feel like we're a little bit behind in this area in terms of the organic crystallizations. And I just listed here a few examples of where do we see these type of crystalline solid solutions. I think people in pharma are very accustomed to amorphous solid solutions, so like amorphous spray, ASD, for instance. So we use it for dry product applications. But when it comes to crystalline solid solutions, it's much less understood. But yeah, you can see here, I'm starting from left here, people in geology they have these plagioclase. This is something I took a picture from the Vesuvius crater. So there's like, different types of elements. So there's calcium and sodium in this case that are substituted in the material. And so depending on how it crystallizes, as it cools down from these lava eruptions, you change in the composition of the material. Yeah, so that's like one example. So, I mean, there are many other examples too. Like I mentioned, steel, and people use semiconductors, and they purposely then add different types of elements to tweak the properties to make sure you can actually control, very fine-tune the properties for certain applications. And going back in history, I mean, we actually had, in human civilization, we had solid solutions all the way back to the Bronze Age. So it's not like we haven't used this before. It's just that we don't understand maybe back then how exactly it works. But we have this, like we have bronze items here on the top right here. as an example. And we also started to see more examples in biology. So there's one recent example showing how certain components of guanine, hypoxanthine and xanthine are actually together forming solutions, and they're actually responsible then for the color changes in chameleons and that sort of things. So it's out there. There's a lot of information out there. It's just that it's not really present as much in an organic area, organic field. So since it's not really known that much, so I just want to give an example of how what actually solid solutions are. And you can think of them sort of like organic alloys. And we are familiar with the stoichiometric crystalforms, anhydride, hydrate, solvate, salts, co-crystals, and so on. These are 1-to-1 typically, or 1-to-2, hemi, di, mono, and so on, right? And solid solutions are very different in the sense that they are non-stoichiometric. So and they can happen through different ways. So one way they can actually appear is through substitution of solid solutions. So that's something they call it for metals, for instance. And see if I can show my where's the here we go. Yeah, so for instance, if you're starting here with something simple, right? We have this structure, this anhydride structure, so one particular product molecule, making it very simple here in 2 dimensions. And then you have a substitution, and suddenly now you have a very structurally similar component that's embedded in the crystal lattice of the host. And of course, as you increase in the concentration, you will see these effects over here, right? But interesting and separate from metals and atoms is that you don't have to have a 1-to-1 sort of substitution. You can actually have like 2-to-1. So you can have like dimers, for instance, that can substitute, depending on how the flexibility and the conformational properties. So you can actually substitute 2 in a crystal lattice and take position like this, or even sitting in different, spatial dimension, or even like bend so that it fit into a crystal lattice like this. Of course, these are examples here, A to F here, where the substitution, the substituents here, actually doesn't impact the crystal lattice notably. But in most cases, there are some significant differences between the embedded foreign molecule here and the host. And so what happens here is you cause these like strains in the lattice. And this is very well known in people working in metals. They have this, we control, for instance, the properties of steel by adding carbon into iron, right? And that produces hardness in different ways that we don't see for iron. So you'll see this sort of like the defects that propagates through the lattice, and it can be like a larger molecule that is embedded. It can also be smaller molecules, so now you have sort of like a point defect towards that center of the lattice. And you can even have interstitial solutions. So here's an example on the right here, an eye, where, like, for instance, a solvent that is embedded, and it just pushes out a little bit on lattice, and it sits, maybe even in voids or in that sort of like pockets in lattice. And importantly here is that every time we have this sort of substitutions or this sort of molecular incorporation, you'll see that the structure is completely unchanged. So if you're doing X-ray and all this, you're gonna see the same thing. You're gonna see your host product, right? XRD is an average analysis over billions and billions of molecules. So you would only see the biggest element of that, right? So you see maybe some disorder in the XRD, but not much else than that. But what actually happens here is because of these molecules that are being incorporated is that you start changing the thermodynamic and the morphological and mechanical properties even of the crystals, sometimes very dramatically. And I want to present a few examples of how that actually works out. So let me start by going into the thermodynamics. So we brought in a lot of interns and co-ops because there's a lot of work here. And we started looking at a very easy system. So I worked with this system before. It's salicylic acid. This is not exactly an API, but for us it's like a simple molecule you can easily buy. It's cheap, and there's only one form of the salicylic acid. And then we added in all these different types of structures, similar compound, to kind of simulate an environment in which would be sort of similar to industrial process. So you would see like these 6 molecules here would be sort of the process impurities, and we would crystallize then salicylic acid. And we constructed this binary TX phase diagrams for all these 6 pairs. And so they're all summarized here on this slide. So starting with salicylic acid, benzoic acid, you have 4-hydroxy and 3-hydroxybenzoic acid here. You can see that these are fairly straightforward phase diagrams. There's the blue one here just shows it's like a second phase diagram for a metastable form of 3-hydroxybenzoic acid. But other than that, they're fairly straightforward. But if you go down to the bottom line here, you have enteric acid, 2,3-dihydroxybenzoic acid, and salicylamide. Now you actually start seeing some complexity in terms of the phase behavior, and gets very, very rich after a while. So I will talk a little bit about that. But let's start with the simple one first. So this is salicylic acid, benzoic acid. And so when we construct these phase diagrams, we don't use 2 orthogonal techniques for it. So one is just DSC to try to get the melting properties and the ranges here, so that you determine the solid is, the liquid is, and you can infer the eutectic composition and so on. And on the right, same thing here, right? And we try to kind of figure out this point right here. So this is the solvus at the eutectic temperature for both sides, because this shows the highest level of solid state miscibility. And we do the same thing using different approach, using a solvent at around room temperature. So in this approach, we determine the solvus, the solid state miscibility limit here at 25 degrees, and we get these points here. But what I want you to see here is that if you're looking at the alpha and the beta domain, that's what people call it in the metals, you have this pretty significant areas right here. So if you crystallize a compound with benzoic acid, for instance, you're going to be right here in the extremes of the diagram, right? So you're going to be close to almost pure salicylic acid, depending how much impurities you have. And what we see here is like during this like little range here, we measure the solubility in methanol water. And as you move from pure salicylic acid to the solid state miscibility limit, the solvus, you can see that the solubility increases from about 23, 24 mg per gram and all the way up to, what is it, like 43 or so. So over about 10% or 11% mole fraction of benzoic acid in the crystal lattice. Right, so this is one solid phase, right? If I'm going across the solvus here, now I'm getting 2 solid phases with the composition of the alpha solvus and the beta solvus. But right here, this is just one solid phase again here. So you can see that there's a pretty significant solubility enhancement takes place. So about 87% up to this. So obviously there's a lot of implications for this as you're working with processing. And sometimes it's hard to measure this because, I mean, one thing you can do is you can just dissolve up benzoic acid in a liquid, right? And then you're letting it sort of equilibrate depending on how long time you have, and then you measure the solubility. But there's also another way you can also, prepare these crystals in advance, either by using some sort of like you can recrystallize it, for instance, and then use that to measure solubility. But you also can like just prepare a physical mixture and anneal it in a vacuum oven at higher temperature to let it slowly blend into each other. And then once you have that final material, then you can suspend it in solution. So sometimes this is not always obvious. It depends on how you actually measure it. But again, if you crystallize this and I introduce pure seeds, you can see that, I mean, if I'm picking a supersaturation ratio here about, like, I don't know, 1.5 or something like that. So maybe my solubility or my concentration is about 35 mg per gram, right? I'm adding in pure seeds in a impure solution. And the effective solubility for that particular condition is actually maybe higher. What actually happens is that I'm seeding with a material that has a lower solubility that slowly starts to absorb and so diffuse into the crystal lattice of the seeds, and eventually those seeds will start to increase in solubility and will eventually dissolve. And we're so used to, I think, in our field to think of everything in terms of solubility always goes down, right? We have form 1, form 2, and always this discrete sort of like understanding. Form 1 always goes down to form 2, and everything, free energy always, goes down. So you always have this solubility decrease. But for solid solutions, it's actually quite the opposite sometimes. So sometimes we see like the solubility increasing rather than decreasing. Okay, and then here's another example that is a lot more complex, right? So as we have this like solubility enhancing effects, they are separate then for different polymers. So as you introduce a polymorphic system, so in this case we're using 2,3-dioxybenzoic acid, it has two forms, form 1 and 2. And as you introduce a binary system here, you can actually have this very interesting type of phase behavior that are very well known in metals and inorganic material, but not so much in organic materials. And so if I'm starting here with the at 1, so this will be pure 2,3-dioxybenzoic acid. Like I have my form 1 and heating up, it eventually melts right here. Okay, now I'm starting to introduce salicylic acid into this. I'm moving left in the diagram. And it only takes about 1% or so. And now suddenly there's a shift here in the stability of two polymers of dioxybenzoic acid. So at this point, I'm moving into a coexistence domains where the two polymers are equally stable and they will persist indefinitely together. Yeah, and if I'm just adding more salicylic acid here, eventually I'm moving into domain now where the previously metastable form of 2,3-dioxybenzoic acid becomes thermodynamically stable. So I have a completely changed the stability relationship of the two polymers by introducing salt solution. And this happens because we are selectively changing the solubility of the material, the two polymorphs. So in this case, there's a much steeper solubility increase for form 1 as salicylic acid becomes incorporated, whereas form 2 here has a much slower, kind of like a weaker dependence on the incorporation of salicylic acid. So yeah, so here's an example of how you can sort of like totally have a shift in polymorphism just based on a second component. So this is obviously important if you're trying to crystallize something in an industrial setting where you have impurities that have this type of behavior, and you're trying to implement a form control in the process. There's also cases here where you can have the opposite, right? So you saw sort of like how the two polymers sort of converge in thermodynamic stability, and eventually there's a shift there, right? And then one becomes more stable than the other. But you can also have the opposite situation. So we saw this for the 3-hydroxybenzoic acid system. And here again, you have two forms, form 1 and form 2. One is pure 3-hydroxybenzoic acid, and it's a monotropic stability relationships. So form 1 is stable, form 2 is metastable at all temperatures. Now, when I introduce salicylic acid here, we see differences in the solid miscibility between the two forms. You can see it a little bit here on the right. It's not huge differences, and it's not that strong differences at around room temperature, but it becomes more evident as you increase the temperature towards the eutectic temperature. And what happens here is it creates this divergence in thermodynamic stability. So instead of having two forms that sort of cross in solubility and thermodynamic stability, they started to diverge instead. So the metastable form becomes even more metastable as you incorporate salicylic acid into the two polymorphs. And it's just listed here, kind of an example of how this actually presents itself if you're looking at solubility ratios. I mean, for people doing solution-mediated phase transformations, so SMPT, you have one form and you're converting to another. I think you're familiar with the solubility ratios, right? So as you're starting here with a pure system, so pure 3-hydroxybenzoic acid, the solubility ratio about 1.32. But then as I incorporate salicylic acid, suddenly, for the same composition here in the solid state, suddenly now the solubility ratio starts to increase a lot until you're hitting the form 2 solvents or the beta 2 solvents. And then you're getting this really weird behavior where it's hitting this high peak here. So you're getting actually enhanced conversion rates as you introduce salicylic acid. And then it drops down again depending on the location of the two solvi. Okay, so obviously salt solutions and polymorphism is related. And if you look back at the literature, you have to sort of really go back like 100 years. And there's some early work by I think Otto Lehmann is the guy that came up with monotropic and anisotropic relationships, and they actually only apply for temperature and pressure for a pure solid phase. Okay, they do not account for composition, which is the third parameter that really determines polymorphism. So we had Rosboom and Bacchus at that time. They showed through different phase diagrams how like temperature and pressure can generate 4 different types of monotropic and anisotropic scenarios, thermodynamic scenarios. But that's not accounting for composition. So if you look in the composition, I mean, you can also use that as a parameter then to alter polymorphism through solid solutions. So for that, we have 2 examples that I described recently, or just briefly. One is the convergence of thermodynamic stabilities. We call that a ostensotropic relationship, and then a divergence in thermodynamic stability, like in the 3-hydroxybenzoic acid example. And then we call that a cryptotropic relationships. So we get in this sort of like looking at solvent solubility, you get in this divergence in solubility versus this convergence in thermodynamic stability or in solubility. Okay, and then quickly here, just before I move into the next area here. So what I talked about here so far was 2 solid compounds, right? So they have, fairly similar melting points, but solid solution formation or crystalline solid solutions, they're not only restricted to 2 solid compounds, you can actually form it with a liquid as well, or even a gas, in fact. So in terms of if you have a solid liquid type of scenario, you're looking at a phase diagram on the right here. So you have a high melting point here of, for instance, the API, and then the eutectic temperature is very, very low, it's subambient. So maybe it's -50, -100 degrees. It's hard to say, right? But when you start using your solvent, you can operate right here. And so you have this liquid phase here, right? And the liquid is here, that's the solubility that we usually measure as a function of temperature. So this is the point here where you measure it. But at the same time, there could be this solidus that appears on the left side, meaning that you have some sort of solid-state miscibility with the solvent or liquid. So you're creating, essentially you can measure solubility and you can measure the composition of the solid phase at the same time. And we have always problems with solvent entrapment in crystallization, and this is probably the most under-researched area in crystallization ever. There's almost 0 publications on this. So it's something that really needs to be looked into more, because we always have to abide by ICH limits. So for instance, ethanol cannot be more than 5,000 ppm in the final API and so on. So it's quite important. And in this study, this is just recently published. So we had a co-op again in the fall, Sam Patil. So she worked on this particular complicated compound, the 763 you see on top here. And she actually managed to measure the solubility and how it actually is as a function of residual solvent content in the same solvent system. So you can see how the solubility increases about 50% at up to 1% of solvent entrapment. So here again, you see an example where solid solution formation can really make things complicated, even for a system where you don't have the typical process impurities. Okay, so let's go into when you crystallize it. I showed you like the thermodynamic equilibrium type of scenarios, the phase diagrams and so on, but what happens when you crystallize these type of compounds in the presence of these solid solution forming impurities? So here's an example again. We're using salicylic acid, and we're adding in 5 of those solid solution forming impurities in about 2% each, and we crystallize it at 40% methanol water. So we're just doing adding in water, and then we're letting it either seed or nucleate through primary nucleation. And then we're pulling samples throughout the crystallization to see what actually happens. And I should mention that all these impurities are andesaturated in terms of their pure solid phases. And if you're looking at the phase diagrams, they're all within the sort of the alpha region, the solid solution domain on the left side in the phase diagrams. So, and this is what happens. So we take samples now. So we track yield versus the impurity level in the solid phase. And in every time we do this, for all the 5 impurities we have in here, we see the same trends over and over and over. And that is that in the beginning of the crystallization, we'll see much higher impurity level than towards the end of the crystallization. There's always like this decreasing, almost like linear slopes going down. And you can take these samples, right? We have solid samples here. So we analyze by HPLC, but you can also do a DSE to see what actually happens to the melting temperature and melting enthalpy. And here you see that, the early samples here have a much lower melting enthalpy and melting point. You can see in this 45 seconds, and it sort of like slowly increases here, just exactly what you expect for solid solution formations. And they're so tightly correlated here that you can plot them on the same, graph, and you can see that the purity and the onset melting point are very much correlated in this way. So what this means is then that we are creating metastable solid solutions early on, and they slowly approach equilibrium as you're crystallizing out. And this early metastable solid solution phase, obviously, because they're higher in purity load, that means they have whoops, that was not my intention. That means they also have a higher solubility initially. So the concept of supersaturation changes when you're working with solid solutions. So, you can't say that the supersaturation is this, as you're starting the crystallization. It depends on the solid phase composition. And also that you're getting more disordered material in the beginning, right? So that's kind of speaks to the non-classical nucleation theories. But all right, so why do we see this behavior? Well, to answer this, we looked at crystallization at different seed loads. So we started at, letting it do, crystallize by primary nucleation, and then we increased the seed load up to about 20%. And then we discounted the effect of those seeds to get like actual corrected plots for what actually crystallizing out over time. So that's listed here. So here again, you can see the 5 different seed loads here. So there's like no seeds up here, and the 20% seed load is down here. And we can also calculate based on our phase diagrams here, and also based on other experiments, we can calculate what actually equilibrium will be. And equilibrium in the solid phase will be close to 1%. So when we do these experiments, we see two things, right? So if we're letting have a high seed load, that means we're going to have growth-dominated crystallization. You can also see that in the Blaze PET data here, we're getting much thicker crystal, more growth, right? And then it just proceed essentially very close to equilibrium. So we're just getting growth there all the way down to equilibrium. But as we are letting it crystallize through nucleation, mostly, then it approaches from the top. So what we see here is that it's the nucleation that drives the impurity entrapment early on. So we're having much higher impurity level in the beginning, and then slowly approaches equilibrium. And this sort of like this change here towards equilibrium takes time. Depends on the compound. For salicylic acid, it's fairly fast, but for other compounds, it's really slow. So yeah, and that just means that nucleation itself we always think of nucleation as sort of like you have your product and maybe your solvent, and it comes together in either there's a disordered state, and then it slowly brings it into a ordered state, right? And then we always think of those two components, but there actually seems to be a lot more than just one component or with the solvent, right? So it's a multi-component phenomena, I would say, nucleation. Yeah, that's a separate talk. But as we are having these changes during the crystallization, we're starting a very high impurity level, we're slowly reducing it. That also means that we're changing the solubility of material over time. So you can think of it sort of like a ripening mechanism that happens. So in this particular case with salicylic acid, you form this dirty nuclei initially that grow into this very thin needles, high aspect ratio initially, and then they started to grow in the more axial direction. So you're getting these sort of like impure cores in the beginning here, and then cleaner outer edges. So solubility is very high initially, and then it slowly drops down as the concentration goes down. And these impure cores then are, as you're approaching the equilibrium solubility of salicylic acid, whatever that is in this particular system, you are making the dirtier domains of the crystals undersaturated. And that means it starts to dissolve back in solutions. So you're creating this hollow, kind of peculiar hollow crystals that forms during this crystallization. So this is I think solid solution ripening or this solid solution effect is quite known in inorganic, but not so much in organic again. And yeah, it's obviously very different from what we call oswald ripening, right? So here we have a solubility that's dependent on the composition of the solid phase. It's not a surface area or size, like nanometer size effect that's coming from this capillary effect that's in the Gibbs-Thompson equation and so on. So it's independent of surface. surfaces, but it's directly dependent on the solid phase composition. Okay, and it's just a quick word here on disappearing polymers, because if you see here in the early stages of crystallization, you're creating a very high level of impurities in the solid phase, so higher solubility, and then can actually lead then to how different polymers can form, and so they can appear and they can also disappear. So if I'm adding in or taking away an impurity, I can then tweak the direction of what polymers becomes thermodynamically more stable. And the two ways this can actually happen. So there's in the first case here, you can have this, what we call again the isotropic relationship. So you have a convergence in thermodynamic stability with the increasing concentration of impurity in the crystallize. And you would then crystallize it here. And then now suddenly you have the metastable form. When it's pure, suddenly becomes the thermodynamic stable form, right? But there's also a second way you can do this, right? So we saw this example in the 2,3-hydroxybenzoic acid, right? We have complete thermodynamic stability shift to a different form. But there's also another way you can do this, and that's through nucleation. So we starting here, so maybe equilibrium is here, but now we're nucleating at much higher level, and then slowly through solid solution ripening, it drops down to this level. So it's possible then, kind of like the Oswald rule of stages, that you have a nucleation that forms for that particular scenario is actually more stable. So from tone will be more stable, and then slowly as the ripening proceeds and you're reducing the impurity load in the solid phase, you're again switching up the stability. You're crossing this transition point right here, and then going down to the previous relationship. So these can then be a thermodynamic explanation for why we see disappearing and appearing polymers. And this is something that we actively pursuing in polymer screening, and we have some great success with it. But with that, I think pretty much over time. So I just want to summarize here and say that crystallization of pharmaceuticals, even small molecules, they always involve other chemical species. There's nothing that is really pure. We have these effects, and they are present at different levels and different magnitudes depending on what the impurity is and the concentration. And we see these salt solutions forming all the time with process impurities. And sort of, I think that salt solution really changes how we think of crystallization. So it changes how you think about solubility, supersaturations, how the relationships with other polymorphs, even morphology changes. So there's a lot of things that are, at least for us, is totally altering how we approach crystallization because of this. And the other thing is we see that nucleation is associated with much higher impurity load, and that we have this solid solution ripening that takes place over time. And and if you compare like this solid solution formations with many other phenomena in crystallization, there's actually, you can give a lot of explanations to how they actually happens, like appearing and disappearing polymorphs, terminal crystal growth, and so on. So with that, I just want to thank you everyone for your attention, and I just want to acknowledge all the people that have been working on this, so within our company and also through the co-ops and the academic collaborations. So thank you so much.
Thank you very much, Fredrik. We have few questions. So the first the question that came out was that, have you tried quantifying the crystal lattice strain upon increasing the impurity incorporation? Example, benzoic acid through Ritman refinement or powder XRD pattern. So that's a question from Luca Russo.
Yeah, it's a really good question. We did try that. I mean, we don't have a whole lot of time to pursue these sort of things. I mean, most of the times we use co-ops and interns to do this type of work. But we did try it. We had one of our crystallographers, he actually prepared about 10% benzoic acid in salicylic acid, which is about close to the solid miscibility limit. And he then put a crystal on the single crystal XRD and tried to determine the actual structure of it and see what is the actual effect here. Can we see anything of benzoic acid in there? And he got back salicylic acid. So we didn't see anything from the benzoic acid changes in the environment of the crystal lattice. So what you have to do is, like, X-ray is not going to be the technology for exploring this. Instead, you're going to have to go into solid-state NMR, or maybe it's Raman or something like that. But we did have an analysis using solid-state NMR for the salicylic acid, antronic acid. I can show you that. system quickly. So that would be this particular system. We could see kind of like the environments in terms of how enteric acid is embedded in the crystal lattice of salicylic acid. We had to label enteric acid before we did that study. But yeah, you can get some information. You can see that it's uniformly distributed in the crystal lattice.
Perfect. Thank you very much. So, Elena, one last question.
Yes, I'm not sure if the chat will stay on after we disconnect. So
Yeah, I will.
it might be go through all of them quickly, maybe.
Yeah, I will copy all the questions that we don't have time
Okay,
to
okay.
and I will send it to Fredrik. So the next question comes from KTH. The great talk, Fredrik. Intriguing thermodynamics. So in principle, by combining solid solutions ripening with Oswald, you should theoretically be able to observe, example, form 1, nucleating, then transforming to form 2, and then grow on them to transform again to form 1.
Yeah, I never thought about using the Oswald, I mean, a nanometer Oswald ripening, but I suppose there's a lot of thermodynamic scenarios that are possible. So yeah, I guess that's possible.
Perfect. Thank you
It
very much.
depends on the nucleation, right? And how you do the thermodynamic treatment of the nuclei, because that's not really clear. There's so many different uncertainties there, but yes.
Perfect. So this is from Michael Zwart, whom you probably know. And I will send the remaining questions to you by email. And if you have time to respond them in case the chat closes and we lose all the questions. So, Elena, we proceed with the last part, right?
Yes.
Just a brief collection of the ideas. So if all the presenters can turn on their cameras. So, well, first of all, thank you very much, particularly from people joining us from the other side of the ocean for waking up so early. And I want to basically ask you, the same with the same order that you discussed. In your field, as you see it, what would be the next challenge to address? So it's a very broad question, I know, but I would really need a brief answer in line with time. Starting from Jerry.
Thanks, Burak, putting me on the spot. There's so many questions, but if I had to pick one, specifically on peptides, I like to know when a peptide is a small molecule and when a peptide is a big molecule.
Yeah. Thank you very much. And also, as we speak, if you have any questions for the panelists, please write it down. I will pass all the questions to them, even if we don't have time to discuss about them. And Joana?
That's a very tough one. I would say, based on the current trend of the big pharma, I would say thinking about new modalities. So I try to highlight the fact that mAbs are very complex, very difficult to crystallize, but there is way more difficult things to crystallize. And the big pharmas, for example, Pfizer is investing a lot of money in ADCs, antibody drug conjugates. There is also other new modalities, so I think this might be the new challenge.
As you see, a naked man joined the talk. So now, Fredrik, would you like to continue on this?
Yeah, sure. I mean, I think I can only agree with the previous presenters here. I think the challenge in the future will be, at least from an industrial perspective, will definitely be in the more complexity of the APIs and I mean, even the small molecules. I mean, you have peptides, you have proteins that are becoming more and more prevalent, but also the small molecules are becoming more and more complex and getting bigger and bigger and bigger. More and more functional groups, more flexibility, and harder to crystallize. Sometimes we have to dedicate a lot of effort to just crystallize a compound that's entering into development. So yeah, that's the biggest challenge right now. So some of these molecules are exceeding 1,000 molecular weight. And so, what do? You have to like approach it very differently. It takes a lot of resources and so on.
Yeah.
Yeah.
Yeah. And maybe a continuation question on this to Fredrik. So could solid solutions be also an asset when you are trying to manipulate solubility? So as you mentioned, there are always limits, safety limits.
Mhm.
Could we design certain solid solutions to achieve a given goal dictated by the manufacturing?
So this is totally unexplored, but I mean, we could definitely, utilize it to a different extent than we have in the past. So there's the one example that comes to mind. So this is Jarka, a professor at Rowan University. So they used paracetamol, and he put in dyes. So he worked with dyes at very like ppm levels, very small levels. But even there, we see the solid solution formation. But in one particular case, use acid fuchsin or something like that. But in one particular case of paracetamol, we saw a completely change in the mechanical properties of the material. So paracetamol is like very big, large crystals, very rigid, right? But with this 500 ppm of this dye, the crystals started crumbling and they were like very, very brittle. So you can absolutely change the properties of material. So mechanical properties you can change, morphology, as I presented, right? And you can also change the solubility. So, but it's the question. The biggest question for us in terms of solid solution formation is what compounds form solid solution. It's really hard to predict this. There's some efforts in terms of what you can do, but if you can like add in something that would tweak the properties, make it more soluble or whatever, you can even stabilize the metastable polymer potentially, right? Then you can use that if it has more favorable properties for processing. But it's totally out and open. So I think that's would be really useful to get some research there.
Yeah. And so if I may then direct the same question to Jerry and Joana, well, what do you think? Could we be so the question is, could we design molecular entities that are still within the regulatory limits to alter, let's say, solubility and other crystal properties rationally, hopefully? So I again pick Jerry as he's my favorite question mark.
You go from left to right in your screen. No, I think almost certainly Fredrik did mention about Jarka work, and I think it's amazing and beautiful how much small levels of impurities can change quite a number of properties. So it's the knowledge, right? I think at the end of the day, if you understand how impurities behave, that you could use that in your favor. And we've done that from using templates to control nucleation. There are adding impurities to the system. There's work also from elsewhere. I can't possibly mention everywhere, but I'm picking up from Limerick, for example, in using excipients as an additive to the system to again control nucleation. But the list goes on. So I think if we understand well the effect of, say, foreign substrates or foreign entities, impurities in the system, we could use that to design and control crystallization processes.
Perfect. Then Joana is the remaining victim.
Yeah, I don't have much to add. I think, what Fredrik already mentioned was kind of a very complete answer, and then also what Jerry mentioned. Yeah, I think in terms of heterogeneous nucleation, definitely impurities, soft templates, they definitely play an important role. I don't have much experience in terms of impurities that can act in my favor because all my impurities they are not really helpful. Yeah, so I guess I don't have much to add on this.
Yes. Thank you. So I'm also waiting for questions from the audience. So please write it in the Q&A. And Elena, would you like to continue?
I think we got to maybe the end of our time. and I can see that people are disconnecting, so maybe it's a good time to kind of like thank all the speakers, thank the organizer, thank the working party of Crystallization, and yes, stay tuned for the next spotlight talks and the next events. So yes, thank you all, and see you very soon.
Thank you. Bye-bye.
Bye.